primary liver cancer. Because they have huge
Algeria,
problems. Huh?
right?
No, not so much that's
Oh, they have
that's
an
Russia.
don't they have an enzyme
Yeah.
that makes them they can't like process
That's part of it,
alcohol?
but the main part is just a lot of hepatitis C.
Oh.
That's the big part of it, is a lot of hep C. So the medical, it's primary prevention doesn't work there. It's getting better with the drugs.
In Asia?
What is it? What is it? What is it?
And maybe just China.
What is it that makes them? You know how they get flush when they drink?
So
It's an
that
intolerance.
particular
No,
population,
they have like
Native
a-
Americans, they don't have, they have a less functional
Yeah.
enzyme, second enzyme in the alcohol. It's called aldehyde dehydrogenase.
Uh-huh.
So that doesn't work as well. So the first step of alcohol is to turn it into an aldehyde, and then it should be rapidly broken down into acetate.
Yeah.
They don't break down as fast, so you get.
Yeah, my cousin's half Korean and he's all like
Mhm.
when all around.
Yeah, no.
Yeah, when he drinks.
But it's funny, I have a friend who looks like, you look up white guy in the dictionary, and he would be there. He drinks, he drinks like that too, so you gotta have something in his background somewhere. Yes, and or Native American, same thing. So contributing factors associated with the development of cancer is aging population growth. And we just talked about this if you were, or one magnitude here, the other. We're just talking about that. As you get older, your immune system becomes less functional. So your immune system is primarily your cancer preventative mechanism, right? And then lifestyle choices. Smoking, physical inactivity, eating a westernized diet. The big ones now are what? So, smoking is starting to go down finally. This is what they call the new what? Physical inactivity, they call the new what?
Physical inactivity?
Inactivity, or they call sitting. It's
Oh,
the new smoking.
potatoes? Oh.
No, they call it the new smoking because they're starting to really link a lot of cancers because of the chronic inflammation and stuff to inactivity, physical
And
inactivity.
if you smoke, you don't, it's not like you can run, you know? Or do exercise.
Well, yeah, also double that if you smoke, but with the advent of computers, how many people spend 8 hours a day sitting behind a computer doing work? And then we don't have time to go out and do this, we don't have anything, so.
Whereas suppose like in Europe, how they have like way different working hours and stuff.
They do, yeah, working hours, they do have this westernized diet, depends, this another one. This is very controversial, they still show the link between the westernized diet, still don't think they've teased out all the exact parts of it, because it's not just some of the fats, some of the fats they found out are healthy fats, are good food, right? Other ones they don't know, so I don't know. What's the best diet, they just did a study.
Mediterranean.
The Mediterranean one diet, so they have a lot of healthy
Yeah.
fat.
They just said Spain is actually the healthiest country because of it.
Yeah.
When it comes to westernized diet, they're not talking about all the preservatives and things that we have.
You're talking about hamburgers, hot dogs, lots of fatty
Like all the
foods,
chemicals.
lots of processed carbohydrates.
That's what I was thinking when it said that.
That's the westernized diet.
Okay.
They're way different.
Cancer causes 1 in 4 deaths in the US still. And for both men and women, the most common cause of cancer-related death is still what?
Lung.
Lung cancer, and that's primarily what?
Smoking?
Smoking, yeah. Big one. And so it's actually going down.
I thought it would be
Lung cancer.
increasing because of like the vaping. Now everybody
Well,
like they
that's what I wanna wait and see what happens with the vaping, because they just haven't done much research on it, because it's not covered by much.
I did see a study that it crystallizes
It
lung.
does do some of the same damage as smoking does, yeah. Problem is, if you're one of the companies, you're gonna say, well, it may, but you can't tell me what it's gonna be long term yet, so they're gonna have to wait until it's all mitigated. My opinion is if you wanted to vape, I would use it as a way to get off of cigarettes, because that's what some people do.
It's not the same thing, though.
Yeah. All right, these are different right here, so let's look at the different ones. Highly developed areas, look at this. Incidence mortality. Australia, New Zealand,
And
lots of cancer, fun place to go, gonna get cancer. Okay, why do you think New Zealand, what do you think might be a contributor to that, to those two countries?
In Australia, we do.
I mean, this is just speculation, I'm just gonna say what I think might be able to come up with possible.
Is
Because
there-
this is just cancer in
in
general.
general, it's not like a specific cancer.
No, this is just incidence
It's
of
the
cancer.
diet, right? That's the only thing I
Cancer
could think of.
cases,
That's
deaths involved in them.
Oh,
No.
because the medicine they don't have?
Well,
No, these are first world
the bad
countries.
part is you look at the mortality and
Mortality is.
Even here, incidence is higher, but the mortality is the same in place where incidences are lower, right?
Yeah.
But
So
the-
think about that.
Wait, okay,
But
The
what's
the
your,
incidence is.
people?
Could be, I don't know, well, no, because that's more, because this is mortality from cancer.
Huh.
This isn't just overall life expectancy.
Yeah.
This is mortality due to cancer. These are the incidents, and these are people.
So
who
what
are-
exactly are you
Because
asking?
Because they're helping them live longer because they have more medicine for it.
More developed countries have better, well, we'll use that term right now, better.
How does that?
treatment.
But how does that explain the incidence?
Yeah, that's what I'm saying.
Incidence
What about
is new.
incidents? Why do you think incidents, right? So here you got Western and North America, we're bad.
Like, why is there more cancer in New
I
Zealand?
think it's the
than-
diet thing.
It's probably the diet.
Oh,
Yeah, that's what I
if North-
said.
If North America,
What, part of it's diet, yeah.
smoking, drinking, Australians are popular for drinking.
Lifestyle.
Lifestyle is gonna be most of, all these are gonna be lifestyle. Because in Australia, you got high drinking populations, you also have a lot of people who spend a lot of time in the where? Sun.
Sun.
So you may have more skin
Oh.
cancer, possibly too. Northern Europe, probably not the skin cancer issue, my guess. I don't think melanoma is a big problem in
I
Finland.
wonder, like Greenland and Iceland, like what?
I think they usually group them in, Iceland I think goes into the Northern European
Yeah.
one. Greenland, I don't know where Greenland actually, I have to look that up. But these are all probably gonna be diet. A lot of heavy what, still smokers in Europe too as well, and Australia and stuff too. So it's all lifestyle, all lifestyle. Southern Europe, a little bit less, they got the, but so, is it just diet?
Mm-mm.
Probably a lot of smokers still in Southern Europe. Central and Eastern Europe, that's gonna be a lot of diet, that's gonna be a lot of, those are still heavily smoking parts of the world. Eastern Europe is just one lot. Eastern Asia, again, probably a lot of these Asian countries have a lot of problems with hepatitis C just in general right now. They also eat a lot of what in Asia?
Fish.
And what kind of fish?
Oh.
Smoked fish. So anything you smoke, where you get those polyaromatic hydrocarbons can cause problems. South America, Southern America. Is it Southern or South America? I think it's called South America, not Southern America. Southeastern Asia, Western Asia, Central America, here they go down. Who has the lowest incidence? It's interesting. It's gonna be, Northern Africa.
Well, it's because they don't live long enough, right?
Those
Well, you could actually tie life expectancy into it, because the older your population gets, the more incidence of cancer it is.
but the way that they eat, like maybe they eat in Africa, like their diets aren't really, and they don't have a lot of
Not
access
processed
to
foods.
smoking.
Right, yeah, so a lot of, any of this may be lifestyle, but it's also could be environmental, a lot of it too there. It could be sun, it could be the heavy smoking populations again, drinking's probably not a big issue in middle Africa, right?
I know that cough syrup is, there's like an epidemic about it, like of people using the,
Oh, the cot?
what is it, no, the one with the codeine.
Which one?
The one with codeine, cough syrup with codeine?
Oh, really?
Yeah, there's like a huge epidemic of it.
Jeez.
In Africa?
Mm-hmm.
What?
Of people using it.
You cannot legislate morality, it does not happen. I'm not gonna drink, I'm gonna do something else. All right, characteristic of cancer, disorder of altered cell differentiation and growth. And it results in a neoplasia. That's what you guys will see it called. Neoplasia means new what?
New growth.
What do I mean by cell altered differentiation? Growth makes sense, right? Tumors are just what?
Just continued growth.
Right, but what I mean by differentiation, tumors are just overgrowth of cells, right?
Mm-hmm.
What does differentiation mean? Or alterations of differentiation?
They change what they meant to do?
Yeah.
Pretty good. I'll find this out with that one.
That makes the cell abnormal?
Abnormal, disshaped, and that's gonna be one of the ones we talked about, dysplasia, right?
Mm-hmm.
What did you guys cover here?
She said a good one.
Like they change what they're meant to do?
Right, and they go back. So if I have liver cancer right now, right? Over here.
That's like.
And I get this big old huge liver tumor, right? That should be great. I got more liver cells. I can go drink it. Woo! I got big liver, right? How come that's not the case?
Because they change.
They undifferentiate. Cells, when they go from embryological, they go through differentiation and they become a particular type of cell with a particular job. Cancer cells' only job is to grow and divide. They undifferentiate and they become non-functional. That's the big problem.
So differentiation means non-functional?
Right. They don't function. So if you have hepatocellular carcinoma, those cancer cells no longer,
They have no purpose.
no purpose. They just want to divide. That's all they want to do. Growth is uncoordinated and relatively autonomous. Again, they're not like they're doing tissue repair or regenerating or responding to stimuli, they're just growing. Lacks normal regulatory controls over cell growth and division. All it is the simple answer is uncontrolled, unregulated cell growth. And it tends to increase in size and grow after stimulus ceases or needs for organisms to meet. In other words, it reaches a point where it becomes autonomous, and it's just gonna start doing what?
What does autonomous mean?
It's just gonna start growing on itself. At first, a lot of cancers need stimuli from your cells, from yourself, from your body or from your environment.
Oh, like nutrition.
That or inflammation or anything, irritation, some sort of growth stimulus, growth factors, and we'll go over it in a little bit. At first, that's the second hit of cancer. After a while, once they become fully cancerous, they don't need any more stimulus or anything else, they'll just keep growing. Prostate cancer is one of them that does that for a long time. At first, when you start treating prostate cancer, you have to have an androgen receptor, and testosterone has to bind to that receptor, and that signals it to grow. So what's the first line of treatment for prostate cancer? You block that anti-androgens, that works for a while. Then the cancer cells lose the need for signaling from the androgen receptor, and they start growing on their own. Okay, cell proliferation is just what? Cell division. So it's an inherent adaptive mechanism for replacing body cells. Tissue repair, tissue replacement. Old cells die and you have to replace them with new ones. You get damaged, you have to fix that, right? That's proliferation. And then differentiation I talked about is specialization. New cells acquire structure and function of cells they replace. That's the ideal way things are supposed to work. Cancer is when both these mechanisms get out of whack. Okay? Leukemia is what?
White blood cell?
White blood cells, but it's white blood cells where?
Non-functional.
There you
In the
go.
blood.
I'm not computer talking to me.
He's trying to answer.
Is that this one or is it somebody else's?
No, it's on.
That's stupid memory thing. All right. What was I talking about?
Leukemia?
Leukemia. It's, why would I talk? I can do
Isn't it the lack of creation of
It's not lack of creation. In fact, sometimes you get bone pain because you get an overproduction of non-functional, undifferentiated,
But it can be both,
non-functional
right?
white cells, huh?
It could be either or, right? It's not only overgrowth.
It's both.
Okay,
Could be
so
both.
yeah.
Isn't it, I don't know, I was just saying, because isn't anemia is
Oh,
that
no, it's
too much blood?
No,
No,
no.
that's
What
not
is
red
anemia?
blood cells.
Lack of red blood
Red
cells.
blood cells.
What is the definition of anemia?
The lack of oxygen that blood can no longer carry.
Good. Lack of oxygen carrying capacity. Good.
Yeah.
There's,
So you
anemia
still have
is a
the red
symptom.
blood cells?
I was just saying because of anemia.
It could have been. You can have lack of blood cells, you can have lack of hemoglobin, you can have, lack of functional hemoglobin, you can have lack of vitamin B12 and folic acid that make red blood cells. Anemia is a symptom of a problem. It's lack of oxygen carrying capacity by the blood, for whatever reason. And you can get that with leukemia because they overgrow and start crowding out the other stem cells, so you can't make enough red blood cells.
Oh,
But they become undifferentiated, unspecialized, and then they start what? So a lot of people with leukemia actually become immunocompromised because they're what?
They don't have the.
Their white blood cells don't work anymore. They've got them, but they're not working.
Non-functioning white blood cells, right?
Right. All right, in apoptosis, the idea behind apoptosis is
Cell
programmed cell death, and the idea behind that is you're supposed to have an unwanted cell. If you have a cell that has DNA damage, when it's gonna replicate, it should do what before it divides?
Die.
It should go through apoptosis, right? And that's where this gets knocked out too. You turn off the
So in
apoptosis.
cancer, it skips that.
That's what the, like, the fat
You
reduction
get all
stuff.
out of whack.
That's what it does, like,
yeah,
what's it called?
yeah.
The colon, the sculpting?
Oh, yeah.
All right, y'all remember the cell
That's
cycle
cool.
from 1306? Everybody's favorite chapter. Well, nobody likes any chapter from 1306. Cell cycle is when cells start to divide, they have really 4 different phases they go through. We'll talk about G0 in a minute. G1 is what? When your cells are in G1, that's what? Normal growth and functioning. Most of your cells are in G1, they're just doing their job. Okay? If they're going to go down the division pathway, you get signals that trigger in G1, you start getting ready to divide, and once you hit that point of no return, you go to S phase. And S phase is the synthesis phase. You copy your DNA. And remember, too many chromosomes, is that good or bad? That's bad. So once you do this, you need to divide, otherwise you're going to have problems.
That you're gonna have like what's it called when kids
Down
are
syndrome?
Well, that's only one extra chromosome. That's only chromosome 21 for Down syndrome. You don't want all this extra DNA in there, that would be bad. If something gets messed up when you get to G2, and you don't divide, you don't get the signal to go to G2, the cell should do what? It should go through apoptosis. You get to G2, growth and preparing for mitosis. This is when you're copying your organelles, you're getting ready to divide. Again, if you have a problem there, something should do what happens? Apoptosis. You have all these checkpoints here.
So interphase is mostly where the apoptosis should happen?
In which one?
And interphase?
It should always have.
It's all.
In
along
every
different
single,
places.
oh, okay.
In fact, sometimes in G2 you don't even go through apoptosis. The system P53 just shuts the cell down.
this is
It doesn't kill it,
Yeah.
but it shuts down the division process. So the cell goes into what's called senescence. It just goes, okay? For some reason we can't kill you, we're gonna try and fix you. So you're just not gonna divide right now.
Oh.
Did you all remember these lovely guys?
Ugh.
Prophase, DNA condenses, what else happens? Nuclear envelope disappears,
The spindle.
spindle fibers attached to microtubules, right?
I don't remember this stuff.
Metaphase, chromosomes
Hesi.
line up across the middle.
Because of the Hesi.
Anaphase, what happens?
They spindle particle away.
They start to split
They move away from the middle.
from the middle, right, into the poles. Telophase is nuclear membrane reappears and the cells begin to divide.
They come together.
True or false?
They split into,
Mitosis is cell division.
no.
Yeah.
Anyone? We got one no?
When they divide, isn't that the,
Meiosis,
like
you're
when they split
thinking?
into two?
Well, that's telekinesis or, no,
Telekinesis.
cytokinesis. Cytokinesis.
Cytokinesis.
Yeah.
Mitosis is not cell division. Mitosis is division of what? The duplicated chromosomes.
Oh, I see what you were asking. I'm
Cytokinesis
confused.
is cell division. Good. What are these two things called that you split apart? I'll give you extra credit points for that if you can tell me.
What was the
Without Googling it.
Chromatins.
Wait, what was the question?
Daughter?
Sister or daughter?
Daughter?
I don't know. I'm asking you guys. What
What
do
was
you
the
think?
question?
So what is the question?
Sister.
These things are called
Daughter
what
cells,
before they
no?
split?
Daughter is meiosis, no? Or wait.
Chromatid.
They're, yeah, but they're either
That's what I
sister or
said.
daughter.
Chromatid.
What kind of chromatin?
I know, it's sister or daughter.
Oh,
Like
sister,
this?
sister chromatids. They're the sister.
Kind
Daughter.
of sexy. Could it
No,
be
daughter.
Daughter.
Daughter's the meiosis.
Oh.
Okay,
Daughter
sister.
cells.
Daughter is daughter cells or daughter, yes. Good.
But sisters means they're splitting.
Yeah. Which one was it?
Remember right now. Sister chromatin.
They're the same.
Got confused.
They're the same thing, they get copies and they're exactly the same, they're just together and they split apart.
And
Okay.
Alright,
So these
And
are
then-
sister, right? Okay.
some cells reset from replication, yet continues to perform as a metabolic functions. Some things that go into G0, they really never divide after that, they go to G0, they go to senescence. Some will go back into G1, different thing, okay? But this is the cell cycle. Okay? Definition is the interval between each cell division. Genetic information is duplicated, this is 1306 review, right? Duplicated chromosomes are what? Appropriate aligned the distribution between two genetically identical daughter cells. That's where the daughter comes in. Checkpoints, this is the cancer part. Checkpoints in the cell cycle, and there are a bunch of them, provide opportunities for monitoring the accuracy of the DNA replication and either kill the cell or fix it. Because what happens if I get a mutation and I go all the way through the cell cycle?
You're gonna copy itself.
And then what's gonna happen? So I've got DNA, I'm copying my DNA in the cell, so here's my sister chromatid. But I had a mutation right here in this one when I made this copy.
You replicate it,
How
you
many
replicate
more
it,
has
you
to
replicate
Or
it.
what?
of the same
The mutation.
But I just copied it right now.
No, but then,
But yeah, then you're gonna split.
And then what?
and then it's
Make
gonna
more
get a new
Go
one.
through the same cycle.
Okay, so when it goes through this, I'm gonna have 2 daughter cells, right?
Or do you get more mutations?
No, you just copy the
This is one, just the same one?
The problem is you guys are right. If it makes it through the cell cycle and divides, that mutation is now fixed in that chromosome. And the DNA, the repair mechanisms are not gonna recognize it as foreign next time. So
So,
next time this cell divides, both of the daughter cells will have what now?
guaranteed.
The mutation and won't get caught.
so then on here for like the, you said that's the cancer point.
Hmm?
You said that this is the cancer point?
Checkpoint, these are checkpoints,
Checkpoint.
these are checkpoints.
So if it gets through, that means it becomes
You make
cancer.
it through and you get successful cell division, that mutation can become fixed in that population of cells.
So is that how cancer can be genetic? Cause I don't understand how it's genetic, unless it's to do with.
You usually have a genetic predisposition for certain cancers. Like
That
if you
just
have a
because your body
mutation,
doesn't recognize it as cancer?
that or you've got DNA repair enzymes a lot of times, like BRAC1 and BRAC2, y'all know what those
Yeah,
are?
those,
Yeah.
those I understand.
Okay, so that's a genetic, so what that means is they're failing to fix these mutations.
And
That's
they'll always
usually,
fail,
yeah.
pretty much.
That's, but not always, but you've got a bunch of other mechanisms, but those 2 predispose you to cancer. Because you're missing those checkpoint mechanisms. That's what most genetic predispositions are. For some reason, you're not able to fix the damage.
Oh, so they can find the checkpoints.
Right.
Oh.
That's what most of them are gonna be, or you're gonna have an increased sensitivity maybe to estrogen or something, or testosterone, or
Yeah.
something like that. Those are they're gonna be. It's not like there's some weird genetic thing.
Like something you pass on a gene.
Yeah, it's not like there's a mutation you pass on that also that becomes cancerous later on. It's gonna be something else.
The mechanism.
It could be a mutation, and that's what it is with BRAC1 and BRAC2. Alright. But it's not like you inherited this, a germline mutation you got from your parents that all of a sudden later on is gonna turn into cancer. There's something else going on, right? Let me jump over these. So checkpoints in the cell cycle provide opportunities to check it. We're gonna talk about cyclins, cyclin-dependent kinases, and cyclin-dependent kinase inhibitors. These are all part of these checkpoint systems.
Okay.
All right? So cyclin-dependent, what is a kinase?
An enzyme.
It does what? Ah,
Eat. Enzymes eat.
No. but
ACEs
it's
have
specific.
enzymes. What do kinases do?
It speaks of something.
No.
They're kind of things? What kinase? You what?
Is it enzymes that act like?
Okay, I'm not gonna go through it because it's actually right there, but I'm looking for it. Kinase is phosphorylate things. Phosphorylation is turning things on and off.
That's
You
right.
phosphorylate something, you turn it on, you dephosphorylate it, you turn it off. This is what all your signaling is, phosphorylation. Kinase are enzymes that act like a gas pedal, improving passage through the cell phases. By phosphorylation of proteins, you turn those other proteins on, which then turn on some other proteins, and then turn on some other proteins. So
But it also turns it off.
that's dephosphorylation. Cyclins, cyclin A and B, bind and activate what?
Like independent.
Cyclin-dependent kinases, okay? So cyclin-dependent kinases, I'm sorry, cyclin-dependent kinases are what?
Enzymes.
Enzymes that phosphorylate other proteins. Cyclins are the ones that come and do what? Turn these on.
Oh.
So these are cyclin-dependent kinases. The enzymes are kinases, but the kinases need cyclins to activate them. Then they go and start phosphorylating other stuff. And
Kind.
it's a cascade. This one will phosphorylate this one.
What's
And then this will go down and phosphorylate this one.
What's cyclin? Is that a,
It's an enzyme too.
oh, it's an enzyme.
So they bind to and activate cyclin-dependent kinase and do this interaction with them. Cyclin-dependent kinase inhibitors do what?
Stop.
These inhibit
Prevent it.
these
The
guys.
inhibit the binding.
Yeah, they can inhibit, they can change what's called conformational change, they'll have them change shape, but they
Does
can
it turn it
no
off or
longer
slow it down?
turn it off. Well, it's gonna depend on how, well, okay, remember guys, you can turn things, slow things down, or you can make a whole bunch of them and turn them off.
That's what I'm saying.
Yeah, it depends on the pathway. In this case, it's gonna be what? Here, you're gonna put the brakes on to stop the cell cycle. If something's gone wrong, and you're going through this cell cycle, these cyclin-dependent kinases can do what?
Stop it.
Turn these guys off, and then they should put the brakes on the cell cycle division, okay? But you got.
And that's anywhere in the cell cycle.
Cyclin A, cyclin B, actually I got a map, it's gonna be G1, usually G2. Okay, various functions of checkpoint. Again, ensuring integrity of the DNA. Did you copy it correctly? Did you make mistakes? Assessing chromosome duplication. Did you copy them, period? Are they two sister chromosomes stuck together? If they're not stuck together, what's gonna happen? They're not gonna separate properly. They're not gonna move into the cell properly. Assessing the integrity of the spindle. If the spindle is not working and you start to separate the chromosomes, what's going to happen?
It's not gonna separate.
They're not gonna separate properly. So all these different checkpoints. This is all gonna be during mitosis.
All of it?
No, different ones, you've got them all through the cell cycle. So right here, cyclin, see, they got a bunch of cyclin. You got cyclin dependent kinase 1, you got cyclin A and B, it's gonna activate here in what? G1 into mitosis. You've got cyclin dependent kinase 2, cyclin E is gonna be where? G1, so you got them all over the place. And again, I just put this in like.
What's the case done for? Is it just like
Kinase.
Kinase.
And guys, don't memorize, just know what a kinase does. What does a kinase do?
Phosphorus.
Phosphorylate stuff, that's what you guys need to do. So it's all just, all it is about what?
Replication.
Regulation and replication. This is replication, but regulation. You have all these different pathways that have to work correctly for all this to work. Okay. So just know those three. Cyclin-dependent kinases, and cyclin-dependent kinase inhibitors and what their job does. What do cyclins do?
Bind?
Turn on the.
They activate cyclin-dependent kinases. What do cyclin-dependent kinases do? They phosphorylate, initiate the cell cycle, right? What do cyclin-dependent kinase inhibitors do?
Turn it off.
They turn that off. That's what you're going to know. And the reason they do this is they've actually, there's some new cancer drugs out there that work through these kinase inhibitors.
So like during transcription and all that, it has no, cancer doesn't have anything to do with that?
No, not really. What happens, okay, that's, what happens, when would you,
Cyclins phosphorylate?
well, there you go.
The
Transcription, what are you doing with
Yeah,
transcription?
it's right here.
You're getting the code for
The kinases.
You're getting the RNA to make that particular protein. That's already, if you're gonna get cancer, that's already a done deal, right? The only difference we get from oncogenes, we'll talk about transcription, okay? So categories of cell types in the body, well-differentiated neurons and cells of the skeletal and cardiac muscle are unable to divide and reproduce, right? Do you hear about a lot of cancers in the neurons, cardiac muscle, or skeletal muscle? No, they're relatively rare, right? Because cancer usually happens in cells that divide. If you're in senescence, it's very difficult, not impossible, but it's not as common, right? Because you usually get damage when they're copying your DNA. Now, somebody goes, say, what about brain cancer? Come on, somebody say it, come on. Is it the neurons usually in brain cancer?
No.
No.
No, it's gonna be the accessory cells.
the tissue.
Like glioblastomas are gonna be the glial cells, right? Parent or progenitor cells that continue to divide and reproduce. These are gonna be blood cells, skin cells, and liver cells. These are ones that are actively dividing all the time.
Yeah.
Do you see a lot of blood cancers?
Yes.
Do you see a lot of skin cancers?
Yes.
Do you see a lot of liver cancers? Not really. Actually, liver cancer, primary liver cancer is relatively rare. The liver's pretty good at protecting itself. Primary liver cancer, we'll talk about the difference.
Oh, okay.
Undifferentiated stem cells that can be triggered to enter the cell cycle and produce large numbers of progenitor cells when needed. One of the problems they're looking at now with cancer is you've got these stem cell cancer cells that start the cancer, the tumors, and then they even change later on.
So there's cells that are already programmed to become.
No,
cancer.
if something's happened to them, they start going through that cancer thing, but they act like stem cells that keep dividing. They also happen to be resistant to chemo for some reason.
So they're differentiated into cancer.
They're undifferentiated,
Oh.
but then there's another different undifferentiation process that even goes above the stem cells. It's kind of weird, they don't know
The
that.
stem cells is when you get the code to become something, right?
Okay, let me back up, so I'll start, what are stem cells?
They're
The
baby
beginning.
cells that don't have a purpose yet.
What, so embryonic stem cells are what? They're undifferentiated cells, right? That's embryonic ones, when the egg and the sperm fuse,
Like they don't have a job yet.
That becomes a stem cell, right?
Yeah.
And then it starts to divide and they start turning into embryonic stem cells. They haven't differentiated. Adult stem cells are gonna be cells you find way down at the bottom of these pathways that have gone down to lineage, but they haven't turned into a particular type. Like hemopoietic stem cells in your bone marrow. Those can turn into what? Any type of blood cell, right? So they're a stem cell. Adult stem cells, when they're trying to use adult stem cells, is push cells. The golden goose with adult stem cells is they're trying to get some cells that are pretty undifferentiated, like basal cells in your skin,
Uh-huh.
and then push them backwards even further. So you-
Program?
To deprogram, to undifferentiate them even further. And the idea is if it's undifferentiated, you give it the right signals, it should turn into what?
Which
Something
what
else.
is supposed
What other cell you need, what other cell type you need. That's the key to adult stem cells, is they're trying to push them backwards. So if you get something like the basal cells in your skin, right? Those are relatively undifferentiated, right? And they divide. You give those right signals, you can even push them back even further. They're getting pretty good at it, they're getting pretty successful with it. They've already done it for cardiac tissue. All right. Oh, here we go. What am I talking about? There's a whole slide on it. Reserve cells that remain quiescent, remember I said ones that aren't doing anything? Until there is a need for cell replenishment, then they kick in.
What does that word mean, quiescent?
Just sitting there, just hanging out.
Just hanging out?
Okay. When a stem cell divides, one daughter cell retains, and this is the key to it, this is the part with the cancer, even cancer stem cells. And this is still theory, a little bit. When a stem cell divides, what happens? This is what's weird about stem cells. One of them differentiates, and the other one what? Stays a stem cell, stays undifferentiated. So the other daughter cell becomes a progenitor cell type that proceeds through the terminal differentiation. So they think that even happens in cancer. That you have some of these stem cells in cancer that they stem cell, when they divide, one of them stays a stem cell. The
Isn't
other
that
one,
like a mesenchymal?
it depends on what cancer it is and everything. Mesenchymal, depends on
Because
what.
mesenchymal becomes something and then the
Right.
other one stays.
You got, yeah, and I'm not, I hated embryology. So I'm terrible at the germinal cell layers, but yeah. The mesenchymal become, I think, you might remember? I don't even remember, I should, I need to learn this. They become, some of them become your organs, some of them become your skin, some of them become your connective tissue. And I can't remember which. The mesenchymal, I think, become your organ. Somebody Google that real quick.
As a what?
Mesenchymal tissue.
So if one stays the same and one is the stem cell, which one's the bad one then? The stem cell?
No, well, they're both bad.
Oh,
But the stem cell, okay, these are just stem cells in general right here,
Uh-huh.
like your hemopoietic stem cell. When it divides, one of them stays a hemopoietic stem cell, the other one starts turning into
Multipotent
a multipluripotent.
stromal cells
Yeah,
that differentiate in a variety of cell types, including osteoblasts,
yeah,
chondrocytes, myocytes, adipocytes.
yeah. So it's your main,
Like
like
everything,
your
like your
skin,
fat
your fats,
cells.
and all those kind of stuff. That's mesenchymal. I guess I need embryo. I hated embryo. Oh. Now you got me all flustered on embryo. So what happens is, even in blood, one turns into, gonna turn into a blood cell, the other one stays a stem cell.
yeah.
They think in cancer it's the same thing. They have cancer stem cells that divide, one starts becoming a progenitor for the tumor, the other one stays a cancer stem cell.
And that's why they grow back?
Right. And that's why they get a really aggressive chemo, because some of those cancer stem cells they think are resistant to chemo. So you
Oh,
kill off the primary tumor, but you can't get rid of these things. So this is the new thing they're going after now.
that
And
is
then-
cool.
So
Stem
they're
cells are
both
for.
not,
Hmm?
they're both not good, they're both, one just growing,
One's,
one's just
yeah,
programming more.
one's like training cancer.
They're trying to kill like the queen, basically, right?
So self-renewal and the potency differential potential, which means they can just differentiate. They have that potential because they're stem cells. They're undifferentiated.
So this might be bad.
Yeah, they think that's why they've had trouble curing something. Because some of them, they think they'll blast with chemo radiation, they think they've cured it, and it goes away and they get refractory cancer later on. They don't know why they haven't been able to kill it. Again, so I don't think it's sort of hasn't been proven yet. So totipotent stem cells produced by fertilized egg can differentiate into embryonic and what? Extra-embryonic stem cell. This thing can turn into what? Eventually it's gonna turn into everything, right? Pluripotent, pluripotent stem cells can differentiate to what? the 3 germ layers we talked about, okay? Multipotent give rise to only a few cell types, hematopoietic stem cells. They can only turn into what? Blood cells, okay? So as you're going down this list, you're getting more and more differentiated even as a stem cell, right? Unipotent give rise to one type of differentiated cell but remain the property of self-renewal. Muscle satellite cells, epidermal stem cell, like talking about the basal layer, right? They can only turn into what? Epidermal cells, right? Trabeculargonium and basal cell of the olfactory epithelium. Y'all remember that in your nose up there?
Yeah,
All those things are constant. Those are the one things that are constant what?
growing, regrowing.
Renewing, right?
Yeah.
If you're trying all these different things here, if you go down this, even though they're stem cells, they become more and more differentiated to some degree.
And by that you mean specific.
So
No?
that's how they take them back.
This can turn into anything, right?
Yeah.
These can only turn into 3 germ layers. And once you get those germ layer stem cells, you get to the multipotent ones, which can only turn into a few hematopoietic stem cells, can only turn into some type of blood cell. Okay? But then you get the unipotent, these are ones that just replace stuff.
Yeah.
So that's
Oh,
how
just
they
replace
try
them.
to turn it back.
Right, so what they're trying to do is they're trying to get to some of these epidermal stem cells here and push them back to at least here. Or theoretically even here. If you want to look at cardiac tissue, you can push it back to what?
Oh, was that what you were
A cell that's only
telling
going to turn
us?
into cardiac cells.
Is that what you're telling us with people with heart attacks that they're injecting
Yeah,
this?
they're
Oh,
able to do that with a new treatment. They get these stem cells, they inject them over the infarct area, and they differentiate the cardiac muscle.
yeah. Awesome.
Problem is, this is the good part. The problem is when it goes bad, right? All right, so embryonic can be pluripotent. Adult stem cells are gonna be used for what?
Tissue.
Generation and replacement, that's their job. Tumor initiating cells, TICs, these are the ones that get damaged and become those tumor initiating cells. And then I talked about what? Cancer
Cancer.
stem cells. And there's not a whole bunch of information on the cancer stem cell. Because it's still sort of a theory kind of thing, it's not universally accepted yet, right? I'll see if I can find some new information on cancer stem cells. But embryonic can theoretically turn into what? Anything, right?
They can turn into any.
In the 3 germ layers, it's hard, I mean, to get to this one, you have to get what? You almost have to get that fertilized egg, because after, see, I hate embryology. How many divisions is it before it starts to differentiate? Do you know? No, it's a few more than that.
8?
I don't know, we're all just guessing. Right? 27? I don't know.
32?
Because if you have identical twins, before it differentiates, that's how you got identical twins, right? The fertilized egg splits into 2, but it hasn't differentiated yet. That's why they're exactly the same. There's no differentiation. Once it differentiates into that, if that cell were to split, it would do what? If it were to split into 2 now, it would probably die, because you've got 2.
Oh, if it does it before.
You want to get it before it differentiates too much. Because if you have one of these cells come off, it was just one of the germ layers, would it ever turn into a full, complete embryo? No, because it would only give rise to one germ. All right. Oh, here we go. Here's your zygote, your fertilized egg, you get your blastula, which is when it can still what?